Good evening, I'm Prabuddha Kundu, the Managing Director of Premas Biotech. Welcome to the Premas Biotechnology, our first in an ongoing series of talks with the knowledge leaders of the industry of today. This is the first conversation of biotechnology talks. We are delighted to have Dr. Howard Levine with us, a well-known and highly experienced leader in the biotech industry. Howard started his biotech journey as one of the early scientists of Genentech and has worked in various capacities in the process development, manufacturing and management at Amgen, Zoma and Repligen. He has been the founder and CEO of Bioprocess Technology Consultants, a leading CMC consulting company which has been recently acquired by BDO in April 1st, 2019. Many congratulations, Howard.
Thank you.
He is presently the national leader in the Bioprocess Technology Group, part of the life science practice of BDO. So, Howard, thank you so much and welcome to the first in the series of Biotechnology.
Prabuddha, thank you very much. It's my pleasure to be here and have this conversation. I'm honored to be your first guest.
Thank you so much. You've been involved with the industry for a very long time, Howard, and what do you think has been some of the very exciting developments over the past few years, especially with respect to, say, process, manufacturing, and we will evolve as we go on through the discussion.
Certainly. I think if you look at manufacturing processes today, they are done at a much larger scale and in a much more efficient manner than we did them as recently as 10 years ago. With the growth of monoclonal antibodies, we've seen a tremendous growth in new technologies for manufacturing. We've seen the introduction of single-use bioreactors. We've seen the development of continuous processing. And more recently, we've seen the introduction and development of newer and different types of products, such as cell and gene therapy.
Wonderful. One of the first things that I have observed over the last few years has been this predominant introduction of single-use technology, as you said. We see the rapid development on many aspects of single-use. It started evolution with the bioreactors, now processing and so on and so forth. However, we see the downstream side slowly catching up. What do you think are some of the things that would come to your mind as perhaps the things that you would like to perhaps keep in mind or sort of look towards the future, if there was something that you could help us with?
I think two of the most significant aspects of single-use technology. One is the fact that it is now widespread and it's generally accepted within the industry. And there are single-use or disposable options available across the entire manufacturing spectrum. So from the beginning of preparing buffers and media to the bioreactors, as you mentioned, through the downstream process, single-use filters, single-use tangential flow devices, even chromatography moving in the direction. Not truly single-use yet, but we have what one could call disposable options, where we have columns that can be used for a limited number of cycles and then either discarded or recharged to be reused.
One of the things we always try and do is reduce cost of goods. And columns as such and resins do play a big role in the cost of goods. Do you believe that there could be possible alternate modes or alternate ways we could go to the root of the problem and perhaps use membranes? Do you see membranes coming back?
There's always been an interest in alternative methods for downstream processing, for purification, but we keep coming back to chromatography because none of these new or alternative technologies have the resolution or the throughput that chromatography has. And even chromatography itself has evolved tremendously over the last 10 or 20 years. We have much more reliable, robust chromatography media available today with higher capacities, more selectivity, higher flow rates, allowing us to process larger and larger volumes of material in a more efficient manner. But at the same time, we do see the development and the introduction of alternatives to chromatography in certain operations. In particular, we've seen the introduction and almost commonplace acceptance now of things like membrane adsorbers as an alternative, especially for removing particular contaminants or impurities that may be present in the process stream.
Definitely, we see virus removal, we see sterile filtration, we see a lot of membranes being used. But one of the questions which really comes to mind, which I thought I would also pick your mind on, is the ability of resonance for two things. One, we see more and more antibodies being used in therapy. So there is a huge amount of load or requirements of kilograms of antibodies, and it's going to shoot up.
Yes.
I believe there are nearly 200 or 300 different clinical trials ongoing, you may correct me, which use antibodies as one of the frontline therapies.
I think you're absolutely right. I mean, to a very large extent, our industry has become a monoclonal antibody industry,
Right.
with new and improved antibody products being developed every day, some of which are requiring very, very large volumes, as you point out. But I think what's also happening is that with the advent of companion diagnostics and personalized medicine,
True.
we see products being developed in more focused niches for more limited patient populations. And these antibodies, while they're still large demands, they're not necessarily going to be the hundreds of kilograms that the first generation of antibodies would require. They're more potent. Mm-hmm. They are focused in more targeted audiences. And so we're still not growing much beyond where we are today. So I think the industry is growing more in terms of number of products than it is in capacity required for any one single product.
And that's a very good point, because that brings back us to the first point that we said, continuous processing and continuous production, because when you're now dealing with more number of products but on a limited scale, you believe that continuous production will soon become the mainstay rather than scale.
I think we're going to see elements of continuous processing introduced in the industry. We're already starting to see it, but I think it's going to become more and more accepted and more popular. Certainly the regulatory authorities are supportive of it. There is a lot of evidence and a lot of data demonstrating that continuous processing can make a manufacturing process more reliable, more robust, producing a more consistent product from batch to batch. It can also significantly lower the cost required for manufacturing. Take, for example, chromatography.
True.
We were talking about making monoclonal antibodies. In the past, we needed very, very large columns to purify antibodies. In a continuous process, you can shrink the column size, which reduces the capital cost and the capital investment for building a manufacturing plant to produce the same quantities of material.
Absolutely. And with the invent, and when you couple continuous processing with disposable improvements in disposable technology, then you have an extremely potent solution. Do you think in all of this there are certain pointers that you would like our audience to know, as if somebody is really trying to introduce single use and go to continuous processing, what would be those 2-3 things that you would tell them to look out for?
Well, I think first of all, as I mentioned earlier, with regards to single-use technologies, it's really been accepted as an acceptable alternative to stainless steel.
Sure.
And so, whereas in the early days when single-use bioreactors, for example, were first introduced, there was conference after conference talking about the equivalence of a stainless steel reactor and a single-use reactor. I think we've come to the point now where it is almost universally accepted that you can achieve the same levels of productivity and product quality and consistency in a single-use bioreactor compared to a stainless steel reactor. So I don't think there's really any hesitation in terms of using that technology. It's more a limitation of scale. Single-use bioreactors are commonly available at 2,000 liters. There is a 4,000 liter single-use bioreactor on the market. I personally don't see a need or a driver to go beyond that. In fact, I think 4,000 may be even bigger than what the market needs in a single-use reactor. I think when we look at the downstream process, because the technologies are not as fully developed or as robust and have not been in use for as long as the bioreactors, there's still some hesitancy, but single-use columns or reusable disposable columns have become very routine, certainly in clinical production and even in commercial manufacturing. We see more and more people implementing them, whether it be in a traditional batch process or in a newer continuous process.
I think what you're saying is that initially there is a great advantage of going into disposables immediately on the upstream side, but perhaps in the downstream side there may be some sort of a hybrid model that you may suggest, which is single use up now and sort of a not exactly a single use in downstream, but a reusable perhaps single use, and then perhaps in the very near future going into a totally integrated disposable system.
Yes, I think that hybrid is a good word to describe where the process is. I think that in developing a manufacturing process, one wants to pick and choose among the best available technologies, and in some cases that best technology may be a single-use or disposable option. In others, you may want to opt with a more traditional reusable, cleanable option. And I think chromatography is really one area where there's a lot of debate over the advantages and disadvantages of one technology versus the other. I think in the bioreactor space, it's really a matter of scale, and in a matter of what you already have. If you're putting a product into an existing facility, if that facility is built out with stainless steel bioreactors, you're certainly not going to tear them out just because you have a new product that you want to use a single-use product for. But if you're building a new facility or if you're expanding capacity, single-use technologies can be a big advantage.
And with that, Howard, thank you so much. That's a perfect point for us to sort of pause for the first of the talks in the first of the series. We are extremely grateful to you for coming and having this discussion. Thank you so much. I hope that we will be able to have you again for many more episodes.
Thank you, Prabuddha. It's been my pleasure to be here. I look forward to the rest of hearing some of the other experts and some of the other topics that you plan to cover. I'd be more than happy to come back and continue our conversation.
Thank you so much, and thank you so much everyone for looking at our channel. Thank you so much. Bye-bye.